dear bookworms, let's dive deep into the bacterial growth and their nutrition this time. So let's begin. Do not forget to on the notification by subscribing. Let's begin. So, what is bacterial cell division? When a bacteria reproduces by having their sources of carbon, nitrogen, hydrogen, oxygen and sometimes inorganic salts. These are the minimum nutritional requirements for them to grow and multiply. Bacterial cell division is also known as binary fission. The bacteria reaches a critical mass in its cell constitution. During this, a bacteria, when inoculates into a human body or into any animal, it starts to gain its weight by multiplicating their DNA strands, DNA molecules, by doubling them, and the circular chromosomal nucleus of bacteria starts to be divided into two cells where a septa is formed and two cells soon gets divided with equal amount of DNA molecules and DNA replicated strands separated into new complementary strands. These two identical double-stranded DNA molecules are formed and resulting into soon separation by cell wall into two daughter cells. And then our bacterial cell division is done. In depth we will dive in later videos. So let's move on to the next one, which is known as generation time. So what is generation time? The time required for bacterium to give rise to two daughter cells during optimum condition is known as generation time. It means when a bacteria is infecting a human body and it starts to multiply, that time which it requires for generating its first two daughter cells and start the infection into the body of the host, that is known as the generation time. It varies in medical bacteria into 20 minutes because it is seen that cultural mediums are provided into the laboratories, and this causes the improvement of the gut condition. The probiotics we eat, those are all good bacteria which are cultured in the laboratories. Then comes the Mycobacterium tuberculosis, which is 20 hours generation time. In this generation time, most often this affects the lungs of the human beings, where the congestions in the lungs, trachea, alveoli occurs. The person coughs or have breathing troubles. This all occurs due to tuberculosis bacteria, which is known as Mycobacterium tuberculosis. Then comes the M. lepra bacilli, which is 20 days generation time. Means once a person is infected, they doesn't even know that they are infected for 20 days. They roam around even without knowing the fact that they are already infected with a bacteria. And this disease predominantly affects the skin and peripheral nerves, where the person have tingling feeling, they have rashes on their skin. This all results into M. lepra bacilli. Then conjunctivitis, we all know red eyes. It transmits from one person to another by the contact of the eye waters wherever it touches, and it all occurs so fast because it has a 19 hours of generation time. It is known as to be happen due to bacterium known as Moraxilla vacanta. So bacterial count. How to understand a bacterial count? Bacteria has two types of countings. One is known as total count and one is viable count. What is total count? Total count is the number of dead bacteria and the number of living bacteria in your body when it has infected you. And it all comes into a phase of bacterial life cycle at the end, known as the phase of decline. And the viable count also comes in the phase of decline, where only the living bacteria are counted. That is known as viable count. Then how does a bacteria grow? Bacterial growth curve, there are 4 stages. So let's dive deep into this 4 stages. Here is the 4 stages of a bacterial life cycle, where the number of bacteria is written and time of bacteria. First is the lag phase. In the lag phase, the inoculation occurs, the bacteria enters the body and it doesn't start multiplication right away, but it's a phase in between. and where the organism adapts to the new environment and starts their necessary enzymes and metabolisms. Then comes the long phase. Log phase, the bacteria starts replicating, and you can see the graph is increasing into a straight line. The cell division starts and exponential increase occurs. The plotted viable count is in a straight line. Soon after that, after the log phase, it comes into a constant phase known as statutory phase, where the bacteria which were old doesn't have much food, while the progeny cells are increasing in number, and both comes into a balance, resulting into a constant state. The progeny cells equals to the cells that had died. This have their cell growth ceases and the exhaustion of nutrition also occurs. This time the bacteria starts to move into another host through the total cell count, where the living cells are present plus the cells which had died, and the viable cell count is also present, resulting into the phase of decline which we have already seen in the previous slide. So what are the bacterial nutritions? What does bacteria needs to survive? So first we have to know what a bacteria body consists of. A bacteria's body consists of 80% of water, and it has sulfur and has phosphorus, and then they have few microelements known as sodium, potassium, iron and magnesium, manganese, and this all occurs in the affecting environment factors too. The environmental factors which affect a bacterial growth are the moisture, oxygen and carbon dioxide. Moisture you could have seen in the previous slide, where it was 80% water a bacteria needs to survive. It means if the water is in a dehydrated place, they are trying to survive, they would die. And what is oxygen? Oxygen we will deep see into the next one. Next slide. Here is the oxygen. So oxygen content or requirement of a bacteria depends into two types. One is aerobes and one is anaerobes. Aerobes have the oxygen. They need oxygen to survive. So I have given a positive sign. Then a negative sign for anaerobes. As the aerobes are divided into three parts. One is obligate. Obligate means always that there is someone having a strict rule. Means over here aerobes have a strict rule, that is they need oxygen to survive or else they would die. And then comes facultative, where they does not only need oxygen, but sometimes it happens that if they do not have oxygen, they will still survive. And then comes the microaerophilies, which means this is a special type. In this special type, we see a bacteria can survive in traces of oxygen too. Campylobacter is a bacteria, which bacteria is an example of microaerophilies. Under anaerobes, there comes Clostridium tetani. Tetani means tetanus, and this bacteria is an obligatory without oxygen bacteria. Means strictly it follows, it doesn't require oxygen to survive, or else it would die if it has any contact with oxygen. Finally, we come to the temperature. Most of the pathogenic bacteria grows in an optimum temperature known as 37 degrees Celsius, because our body temperature is that, and that's the most suitable place for a bacteria to grow. Here is a small assignment for you to solve. Generation time of Mycobacterium tuberculosis is about A. 20 seconds, B. 20 minutes, C. 20 hours, D. 20 days. Comment below the answer, and this is a reference from Applied Microbiology of B.SC Nursing, and the previous video's assignment answer will be given in the comments below. Thanks for watching. Do like, share and subscribe.